Yeah, I'm the stand-in for Bruce Mundy today, clearly. I don't see a whole lot of other fish people on. This is another cuskeel, and that's in the family Ophidiidae, I think. And I'm going to have to, it's got bifurcated pelvic filaments there.
Oh, yeah.
So that's going to, I think that's going to put it in the genus Spectrunculus, but that's going to be my guess.
That's a good enough guess, Chris.
Spectrunculus.
And
Tina says there was something alive to the left.
Come on out a little bit, video.
Oh, yeah. A brittle star?
No, it looks like it might be a sea.
Oh, maybe a sea star like the previous. Yeah, looks like
no. I think you're right. Brittle star. It's a brittle star.
Oh, yeah, brittle star. Now, is it like the one we saw before with the purple center? It looks
No, I think it's different. It's more red.
Yeah, but the center is darker. You see?
Mm-hmm.
And the previous
Hi,
one.
it's Scott. What was just below that opioid, if you're still there?
Okay, what's right below that?
Oh, yeah.
Yeah,
Shrimp. And I see some two little white amphipods to the left, in the upper left corner. Kind of come up to the middle of the left. I think the two little white dots.
Yeah.
You see on the left?
Gosh, really tiny.
They are tiny. But they kind of look like the amphipods that stick onto the hydroids and other things.
The brittle star.
Should I get us moving toward that last waypoint?
Photogenic brittle star.
Where was that now?
Yes, please.
Should I get us
Close
moving
up
toward that
if
last
we
waypoint?
can. Yes, thank you.
For those of you who are just joining in, we are on the NOAA ship Okeanos Explorer, America's ship for ocean exploration, and we are coming to you live from the Mariana Trench National Marine Monument, and we're exploring a never-before explored ridge, which we have dubbed Stegosaurus Ridge because it looks like
the plates on the back of a Stegosaurus.
Yep.
Anything else here?
My name is Shirley Pomponi. I'm a research professor at Harbor Branch Oceanographic, Florida Atlantic University, and the Cooperative Institute for Ocean Exploration, Research, and Technology. We are going to...
Video.
Yep, we can look at that. And Patty?
Yeah.
Patty is the geo science lead, geology science lead, and I'm from the University of Hawaii, the Hawaii Institute of Geophysics and Planetology, and we are out here
so I got a saddle to my starboard.
looking at a north-south striking ridge that is at about, right now we're at 3,085 meters, about 2.5 meters off the seafloor.
I think big, slow moves are appropriate, right?
Maybe we should introduce the rest of the crew here in control room.
Yeah, you know what? We have not since they came in
for a
after
while.
lunch.
Our ROVs are being operated by a very capable crew from the Global Foundation for Ocean Exploration. And when they feel comfortable, maybe they can introduce themselves.
So this is Josh Carlson. I'm a mechanical and software engineer for Global Foundation for Ocean Exploration.
Wave to the camera, Josh. There you go. That's the one in the middle there.
Okay. Who do we have on nav?
Navigator is Dave Casagrande today. I'm an electrical engineer with the Global Foundation for Ocean Exploration.
And he's a dinosaur specialist also.
I know the correct spelling of Stegosaurus.
And that is a good thing.
Okay, who do we have? Who's our co-pilot?
Lasers are on.
Oh, wait, he's busy right now. Okay, co-pilot.
Co-pilot, I'm Don Libertori, also with GFOE, and I don't have a clue how to spell Stegosaurus.
I'm not going to say anything. Okay, and in the far right is Tara Smithy, who's our videographer, also from GFOE.
You can't see her on screen, but she's doing a great job.
And in our back row, sitting right next to me is...
Sorry, bearing 020 at 0.1 knots.
Videographers, and I'm also from the GFOE.
Thank you.
Wonderful. Thanks very much. You guys are all doing a great job. Ooh, look at all that.
Hydroids on the top of this squat lobster. So Chris was telling us earlier what this was. I think it's the same...
Munidopsis.
Yeah,
Munidopsis.
I'm pretty sure it's the same animal. He's got his back
individual,
feet tucked under. He's got 10 legs, I think Chris said. And he said that it tucks its back 2 legs underneath its tail end when it's sitting there hanging on to food.
I'm getting a lot of credit for Scott's comments.
Oh, my goodness.
When he talked about the legs.
Oh, my goodness.
In any case, I'm pretty sure this is Munidopsis, but Tim Shanks is going to try to go and dredge up some of his information and confirm that.
Is it dinner time for Scott?
Something that we're calling Munidopsis species or Munidopsis kensmithi or something like that. One of the things you can tell for this particular genus and this family is, unlike some of the other squat lobsters we've seen, this is a pretty big one. A lot of these are white, most of them, I guess, are white like this, and they have rather massive front claws. You can see those they're sort of short and stubby and kind of massive claws right there in front. And if you remember some of the other squat lobsters we've seen, they can have sort of really long, delicate-looking pincers, but not this guy. This guy looks like he's a bone crusher.
Well, thank you, Scott, for having told us, and Chris for having corrected me for saying that you had told us. Scott may be off making dinner.
No, he's there. Credit for Scott's comments. He's usually really good on his comments, so yeah, I'll take credit for him.
Well, he
Chris, I'll give you a little bit more. We moved out from now. We can't see it, but Patty, you did ask about there's supposed to be 10 legs. Yes, the 4th leg was missing. You can see the socket hole where it's pulled off or something.
Yeah, I wondered. I did wonder about that. Thank you, Scott.
So, Scott, any reason why these are white, but the ones we've been seeing in shallower water are more orange or red-orange?
Yeah, that's a terrific question. Obviously, there's no light down here, and so, color may not operate in the same way it does in shallower water, but those depths of 300, 500, and 600 meters we've been working at, there's also no light. So I don't know if that's a reflection of the prey that it's feeding on or some other accident of evolution and what it's related to. I'm not certain. We'd have to hear from some of the crustacean biologists if anybody's done some studies on the pigmentation of the cuticle.
There's a shrimp in the upper part of the screen. And the white stuff the white rock we're looking at is just the exposed surface of the sediment, and it has pretty clearly it's got manganese oxide, which makes that black coating on portions of the sediment. Now, why it happens to form on some
and not on
parts of the rock and others could be related to the composition of the material that's making up the exposure. There's something moving through the water, and it's got legs.
Tumbling.
It's got legs.
It's tumbling.
It's tumbling, but it's got legs. Pretty sure it has legs. I saw little tiny legs. Yeah, see, it's trying to swim. It's really hard to get a zoom on this guy, though. He's moving.
He's like,
yeah.
Go ahead.
Paddling like crazy.
He certainly is.
It's like an amphipod.
Yeah, it does.
Oh my gosh.
Yes.
Teeny
tiny amphipod.
Oh, just swimming for
struggling like crazy,
all
probably
it's worth.
in the prop wash, maybe.
I don't know.
It's just moving with the current.
Just moving. It's a very strong current.
Yep.
Oh, thank you, pilot. Thank you. That was fun.
All right.
Nice photo.
It was a nice view of an amphipod, and I just thought I would tell the listeners a little bit about amphipods.
So if you keep your head
many of them are scavengers.
To our
and we talked about this when we were in our
direction we're going,
dive to the
keep
trench.
my head into the current.
And there are some studies that suggest there are 2 groups of amphipods that are scavengers. One that are like blowflies and kind of stay close to the bottom, and another that are more like vultures and stay way up off the bottom, hundreds of meters off the bottom.
Yeah,
but both are kind of drifting around looking for odor plumes, and then we'll swim down to wherever it is that's releasing the odor, presumably dead fish, etc.
Thank you, Scott.
We still have
I'm
3-0
looking at that teeny tiny little guy. It looks just like some of the ones that we saw on the Schmidt Ocean Institute's research vessel Falkor down at 10,800 meters or so.
No, like
These guys were feeding
at point one, we can stop zoom.
Sure. An extended pipe that had a mackerel wrapped around it that some of my colleagues had put on a. Oh, wait a minute. This is a shrimp? This looks different. It looks like a shrimp. So the landers that we put down in the deep part of the trench saw same sort of critters. Okay, what is this? Scott, can you tell us what this is?
Whatever it is, it's swimming madly.
Just like the amphipod.
I'm thinking the video is freezing a bit, but I think this might be an isopod I think it's an isopod.
Huh. Okay.
So isopods are flattened dorso-ventrally, right, top to bottom, whereas amphipods are flattened
horizontally,
laterally. Is that a fairly good?
Yeah, that's a good general rule.
Yeah.
It's not entirely true of all of them. You know, there's always exceptions, and so there are some groups that within each of those that don't fit it. But in the event log, Tina and Les are wondering if it's a Teneiid, which is fairly unusual, at least in the general mind's eye, in the mind's eye of the general public. But it's another crustacean closely related to the isopods and the amphipods. They're all what are called paracarida, which means they all have this characteristic of having a brood pouch. The females have plates below the thorax where they gestate the embryos, and they keep them there until they turn into juveniles.
Ha. Fantastic. Yeah, the amphipods down at 10,800 meters are about a foot long. And the folks put they were looking for some bait to use to videotape them on James Cameron's dives to the New Britain Trench and the Challenger Deep in the Mariana Trench. And in the New Britain Trench, they didn't have the fish to use, so they basically attached a chicken from the galley to the trap.
Okay.
We have a barnacle.
And what are those things called?
They were extended as you
there are legs.
And?
Siri.
Very good.
All right.
Very good.
Two
I'm
barnacles?
proud of you. Yes.
2 barnacles. Now, do they bud off?
Oh, that's a good question.
And I don't know the answer to that. Or did they just happen to land in the same spot?
I don't know.
Because don't they have a pelagic stage?
Yes.
How bizarre. Pick the same rock. That little white thing to the left of them, is that a tiny star?
I don't know. It looked like it, didn't it?
Yeah. Just to the left.
So just a comment on, the 2 barnacles here. Barnacles have separate male and female individuals, and so it's possible that, let's say one of those was a female, that it would be releasing a pheromone so that the pelagic larva might detect it and settle next to it so that they could then reproduce in the future.
Oh,
okay.
That is such a cool story. So back to the chicken. The chicken got eaten in like 5 minutes by, must have been 40 or 50 of these enormous amphipods.
Oh.
And
amphipods or isopods?
Amphipods.
Really?
Yeah, they were the more roundy guys. And there is video on the National Geographic website, and people can go and look at that from James Cameron's dives. It is sped up a little bit, but it's still impressive. So all that came back were the bones and a bunch of amphipods that they trapped. Oh, look, there's a holotherian. There's a red holotherian.
Oh, wow, that's nice, huh?
Splendid. I do like the animals.
Come on in, video.
I'm going to be a convert by the end of this cruise.
Oh,
very nice.
Everybody
Beautiful.
likes the animals.
Very nice.
Okay, my constant question.
I know what you're going to ask.
Is this the front end or the back end of this individual? And how do you tell?
Front or back end?
If we can get it to swim in the water column, we can tell.
Or if it poops.
Yes. Now, that is a four-letter word I understand we are permitted to use.
I'll say poo.
And it is sitting nestled among lovely little pebbles of volcanic rock that have been rounded through the ages. We have no idea how old this stuff is.
Possibly grazing off, scavenging off stuff that's on the that's
on the surface of the rocks.
Yep.
Sure.
Clear.
It's interesting. It prefers the fresher surface and not the manganese-coated surface.
Yeah, I must confess I have no idea which was the front or
to
the
east, so maybe one
front end. But Tina seemed to think that the lower end that we were looking at was the front end. Yep, and Bob Carney says the same thing, anterior end.
Awesome.
Good to know.
Clear video?
Yep, clear.
Okay.
Go ahead. Thank you, Pilot.
It's another.
There's a brittle star to the
left. That's okay. Yeah, we can keep. And another. What do we have on the top of that rock? Another one, perhaps.
Oh yeah, maybe two. Definitely more than one. Okay, so we are headed. Our heading is coming around to 16 degrees.
Go ahead and video.
16, 17, 18, 20 or so. And the rocks are fractured.
There we go.
You can come in tight. Go for it.
Oh, yeah.
Yep, very
This is
pretty.
very similar to the earlier ones we've
There's some of
seen.
those
These pink-colored brittle stars.
with the spines. Yep.
What gorgeous video.
Yep. Annie is sitting next to me, and she's multitasking. She's doing video cutting, and she's my spotter also. Every now and then she'll catch things that are on the corner of the screen and point them out to me.
And elbow Shirley. Hey, look! Oh, these are beautiful. And this one is moving its arm, setting it down, and little tube feet will attach. Look how thin the ends of these arms are. As it lifts it up, drapes it over the rock surface.
Okay, we come to the right. Just a touch, I'll get an establishing of both.
Thanks, pilot.
All right, when you're ready, video.
All right, a couple more seconds. All right, clear.
So our orientation, our heading is about 30 degrees, and we're looking at rocks that are dipping down to our right at a fairly steep angle. Just keeping track of the structure.
To the right or to the left?
Dipping down
to the right?
To the right. Yeah, and maybe, well, we're turning around now.
Yeah.
So we're headed.
Okay.
We're turning back.
DVL really doesn't like this terrain. Huh?
It's hard to keep track
Yeah.
when we're spinning around like this.
Does it?
Yeah.
Interesting. Well, if you get too low, it'll drop out.
Oh, maybe it's not.
Yeah. If you get below like a meter or like
you're
half
in the blanking range. Okay.
Yeah, it's just how the DVL
If
is.
folks want to take a look at this,
two
the
beams
second
are too close, then.
view, the second feed that you have available to you, you can see the ROV on the seafloor in the view from the Sirius vehicle, which is on a tether 30 meters up or 30 meters away. The tether is 30 meters long from the remotely operated vehicle, D2. So we're looking at the top of a ridge. It's oriented north-south. That is about 70 kilometers west of the Mariana Trench axis. We're at a depth of 3,091 meters right now.
Weather's calming down. The stern thrusters seem happier.
So what you're seeing on the seafloor is a volcanic clastic material that has been erupted. We don't have the age of this.
Deep zoom of this guy. Is that a sponge? Or
Looks like we have something on the seafloor out there. Looks like a sponge. Surely it's another one of those sponges we saw earlier.
Does look like that.
Looks pretty healthy.
Okay,
Yeah, it does look like another.
This is another of these Semperella. And you know, if we're looking for some biologics to collect, I know I'm a little bit biased here, but this wouldn't be a bad one to collect if other people would agree.
Do we?
That's a good point.
Is this something that we, for which we don't know the species? We suspect it might be new.
Now, if we do that, should I stop calling it?
We don't know the species of any Semperella, I think.
Really?
I'm trying to think of whether Konstantin Tabachnik in Russia is the expert who actually described this genus, but I don't remember whether or not he actually described a species. He must have.
Well,
maybe there's only one. I'm not sure. Maybe there's a few others.
I'll have to check the system.
Yeah, copy that.
Tina Molitsova is saying that it is okay for the sponge as a sample.
And
she's unsure.
I've already asked the pilot to get in position.
Roger. Okay, great.
So are we going to want to cut a section of this off?
Do it part of the guy, or?
And then is it going to be squishy or brittle? That's
how much of the sponge and what texture?
I would suggest that we get the whole thing because the base of it is diagnostic.
Yeah, copy.
And it could be fairly brittle. So we might be. I don't know if you can pick up the rock or not. That it's pick it up on the rock.
Well, it's basically pilot. It's basically going to be sitting on pebbles. I don't know if you can put the claw under it.
Let me comment on this. It will not be brittle. It's a soft sponge.
Okay.
And he'll be able to sort of tear a piece off of it without any problem at all. Also, I misspoke. It looks like, according to the worms database, there's 11 species known throughout the world. But obviously, since no one's really done a lot of work here in CNMI, this could clearly be a new species, and most likely is a new record, or I think at the least it would be a new record.
So, Chris, we don't need any of the base? As long as we have good video.
How big is this going to be? If he could get the whole thing, that would be great. But will that fit in a box, the whole thing?
If it's squishy, I think we should be able to fit it.
Yes.
Looks like it's about 15 centimeters high.
Yeah.
The issue might be more what it's attached to.
Well, if it would fit in a box, the whole thing, I mean, then it has a much better chance of being described accurately if it's a new species.
So pilot, it might come off fairly easily. It might rip off the bottom fairly easily.
All right, copy.
Pilot, this is Patty. I suspect it's attached to little pebbles.
You might get some cobbles stuck in these spicules. The basalia, which is what those spicules at the base are located, or what they're called. But it's not going to be attached to a rock. It should just come up like with some roots to it.
Yeah, I agree, Chris. This thing, the previous one and this one look like they're sitting in pebbles, and I agree with you. It'd be easy to get it off if it's soft enough, that in particular. So it could be about 20 centimeters tall, 15 to 20, I'm guessing. It's about maybe 7 or 8 centimeters in diameter at the top. As this is the only living species that we've seen up here,
try to get a shot of this video. Come on in.
Wow, living sponge. We've seen lots of other live
hydraulics
things.
are
enabled.
difficult
To,
and
oh, well,
the port bio box is ready to go.
Very nice.
Yeah, you can see the base very nicely.
Yeah, you can see it's really nice, yeah.
Right on the base.
So what's great about this specimen is if we can get this to Constantine, given that he is the one who described this genus, we got somebody right now who could really work on
look
this
at all the
and tell us what it is.
Yeah, look at all the filaments.
But I'm sure it is in the genus Semperella, 99.9% certain.
Yeah, Chris, do you see in the lower left-hand side of the screen, as we're close now, it looks like there are all sorts of spicules spread out all around. It does look like it, doesn't it?
It's like a little, like a little bird's nest.
Yeah, so the Semperella that Constantine described was the type specimen came from sediment, if I'm not mistaken.
Okay.
I think that's correct.
Yeah, I'd like to keep it up
And
just because
all of
we're
these
moving.
spicules coming out, these basalia coming out all around the sides are really characteristic of that genus, I think.
Yep.
Whoa, well done.
Nice. Very nice.
Very nice.
Oh,
very
fantastic pilot.
Maybe you
Very
should just
nice,
put that in a
Josh.
block
Yeah,
rather than
that's what I'm
spinning it.
thinking.
Oh, I'm not going to look at it. So
Yeah, we don't
And
need
you
to
can
just.
shake off
You're
that
ready for the
Yeah,
draw.
it's
sponge is
okay.
pretty tough. You know, that's no
Well,
problem doing that.
well, the sponge is tough, but it's not in. You know, it may
tilt-
not be
get
in
my
there
main
really tightly,
Tilt up,
so we don't
please.
want it to fall out.
and then we can open
So-
the drawer.
And we already
Alright,
have
that's
some
good. So
really
open up the
good
drawer, please.
video.
Okay. Drawer open.
So we're just going to put it right in the box.
box. It's beautiful, Josh.
It's
Thank
going
you.
Port, right?
Yep.
So we have a rock in the starboard box, boxes, and this one goes in the port. Forward,
Perfect.
port forward.
Port inboard.
Very
Clear.
nice. Okay.
Port.
Drawer
So
closed.
Shirley, I think it's up to you, but I think a lot of this thing could be dried, and maybe if you take a small aliquot for alcohol and DNA, you know, a reasonable size, and dry most of it, because Henry Ricewig dries most of the specimens that he gets. That would save a lot of alcohol.
Okay.
Someone turned off my hotkeys here. I'm going to have to do this manually.
Okay.
Oh, do you
Let's
need me to
see if
stick
we
here
can
for your...
see anything else out there.
We're doing pretty good on alcohol, though, Chris.
You got
gonna
it?
get
We're
us
being
moving again.
very conservative.
Yes, I have it dropped on you.
Oh, we're not moving right now?
Nope,
my
should
camera.
just
Great, thanks, and great
you good on
collection.
hydraulics?
Thanks. There'll be some. I'm hoping there'll be a couple people very happy with
Bridge,
that
ROV
specimen.
nav.
There
Hydraulics
are
off.
already a couple people really happy.
Another ship move, please. 50 meters, bearing 040 at 0.1 knots. Good copy, thank you. Thank you, Bridge.
Nav, what was that course again?
040.
040, roger.
Chris, did you happen to look in? Oh, you looked in the worms database to see what the distribution was of the Semperellas. Were there any reported from this area?
I didn't. I looked to see how many species there were, but I'll go ahead and take a look now, Shirley.
Okay.
Shirley, have we done a zoom on any of these little white patches to see if they're animals sticking out?
I don't think we have.
Okay, let's try that.
And we are still on schedule.
Good.
Pilot watch lead.
Thanks.
Go ahead.
I wonder if you could just zoom in on some of these white patches just to see if there are any, these may be burrows, see if there are any animals that we can see kind of in the center of the patches sticking their heads out of the ground.
Copy. Come on in video. Like this patch here?
Yeah. Yeah, maybe this or the one above it. We might be looking for, radiating trails on the seafloor.
Some
I don't know.
sea stars in there or things
I
that are
wasn't thinking
burrowing.
sea stars. I was thinking maybe these are the actual animals within the burrows, or see the holes in the burrows themselves.
Come on in.
Okay.
Yeah.
Yeah, like right there. That just looks to me like it's a burrow. I don't know. What do you think?
I don't know.
I don't see anything coming
on the
out.
surface. I
Nope.
don't see anything on the surface that would
Me either.
indicate that there was anything under there, anything big under there. I'm sure there's lots of,
But the texture of the
Polychaetes,
material
bacteria,
around the
crustaceans.
center of that patch
looks like it's
close to it. Is a little different, a little coarser than the white area around the
come back in.
outside. I can't tell whether there's a pit in the center of that and a hole. There's something right in the middle, but I don't know what it is. Doesn't look like it's an animal's head.
Yeah, it doesn't to me either.
Okay.
Okay. Thanks, pilot.
Thank you, pilot.
You're welcome.
Well, so much for that idea.
It's okay.
I still think that's what these are.
That was a nice grab, digging it out.
Thanks. Yeah, I think we grabbed something with that type of base in a similar way last cruise. I think it might have been Carl who did it, and you could just basically dig in your fingers and you just grab all the...
Yeah, it's loose sediment there.
Yeah,
right.
and just come up
Bust
with
it up a little bit, huh?
The only problem is you don't have a good grip. But
well, the little fibery things are pretty tough.
Are they?
Yeah, it's like glass fibers.
Yeah. Someone was telling me they were the inspiration for fiber optics.
Is that right?
Yeah, I don't remember who told me that.
Well, as we move along the seafloor, what we're looking at
Right, Don, we're halfway
is
to
the top.
our next move.
Well, we're going to be looking at a sea star in a moment, or a brittle star. Brittle star, I think. Yeah, that's a brittle star sitting on the top of what we are calling, just informally, Stegosaurus Ridge.
Come on in, video.
About 70 kilometers west of the Mariana Trench, in the Mariana Trench Marine National Monument. Now this one looks a little different from what we've been seeing before. It doesn't have the extended tube feet that we saw in the earlier brittle stars. Has a different sort of body. And so, looking at our chat log, let's see if Scott France can tell us what this is. It is moving across the seafloor. These things can go really fast, folks. So if Scott is out to dinner, which he might be, yesterday he was fixing dinner about this time, as he's calling in from...
Nope, it's past dinner. I was just thinking about it. Bob Carney and I both think it's possibly the genus Ophiomysium, which is a very common brittle star on soft sediments throughout the deep sea.
Oh, thank you, Scott.
Okay, video, if you want to get established.
Thank you, Paul. Sure.
We have lasers on, Don? I think we do. Yeah.
Yep.
We're good.
So the little red dots are 10 centimeters apart, folks. That gives you an idea of scale for this brittle star. That's about 4 inches. And I think we see, ooh boy, something swimming in the water column really fast.
Hey, Chris, I was just looking in the Systema Peripheral, and according to the description there for Semperella, distribution is Indo-West Pacific, depth from 90 to 4,700 meters, mostly from the Indonesia area, so I'm not seeing anything from this area.
Okay, I just finished looking at each of the 11 species individually, and there are none that have been described from CNMI. As you said, there's some in Indonesia. There's one from the South China Sea.
Right.
There's 2 or 3 from New Caledonia, one from India, and a couple other places, but
Philippines,
not from
maybe.
CNMI.
Yep. Okay, so at the very least, new record of occurrence.
Yeah, absolutely.
We're looking at a seafloor that is covered with small to medium-sized volcanics, just pebbles, pebble size, so things about the size of an egg to smaller, and then the larger individual rocks are cobble or boulder sized. And all of these are, we have no idea what the age of these rocks might be. Anywhere from 50 million years ago to maybe some of it may be very recent.
Go ahead, watch
the sediment, the fine stuff. Beautiful shrimp.
1 second.
Clinging to this rock. The rock has a manganese oxide coating on it.
We are on schedule to make that end waypoint. 5 minutes remaining, yes.
So they have almost an hour left.
And we have about...
So, when you're good video.
So Chris Kelly, this shrimp has really long antennae. This is like something you described earlier.
Roger.
Yeah, so I think this is some type of nematocarcinus. The legs aren't as long as one we typically call nematocarcinus tineurostris.
Set on top.
But I think it's probably in that genus.
Come on in again.
Good eyes.
That guy's strange.
Yes, he is.
Looks like a tube worm.
Shirley, what is this little guy?
Oh, that's... Oh.
What is it?
I'm looking.
What is
I bet
it?
it's another sponge.
That's what I'm thinking. That's what I'm thinking, too. Oh, my gosh.
Sample.
This is on a loose.
I'll bet Les will write in the event log. This is kind of reminiscent of some that we saw up in the Aleutians,
Really?
diving deep by some of the trenches there. The Aleutians,
Really?
diving deep by some of the trenches there.
Huh.
Wow. Is it on a loose rock by any chance?
No, it is not.
Poo.
It is on a very large rock.
Wow.
It's beautiful.
Looks like kind of like a dandelion, doesn't it?
It does.
What are they thinking
Oh
it
my
is?
gosh.
Sponge.
Oh, a sponge.
Yeah, it's a sponge.
Wow.
Wow.
Right, it's a sponge.
I don't know. Wow. Is it possible to get any closer in? Can we zoom any closer in?
Is that was max?
That's max zoom?
I can move closer.
Okay. Now, is there something down below it on the little pebble in front of that big rock that has a red tip?
That was the shrimp I was looking at.
Oh, that was a shrimp. I'm
yeah.
sorry. Yep.
Yeah.
We already saw that. Yeah, unfortunately, that is a big rock.
Oh, it is a big rock.
Come on in video.
Yeah, I've never... Wow. All right.
Oh, wow. Beautiful video.
Oh, my gosh.
Oh, look at that translucent tissue around the stalk.
Wow.
And the pink inner part of it. Wow. That's really incredible. Now, Shirley, is it the angle I'm looking at it, but it looks like on two opposite sides the arms are longer at the top of the sponge?
I don't know. It might just be that they're extended out, but they're not.
It's hard
I mean,
to tell.
Are they just really long spicule? I don't know. Yeah,
Shirley, so some of those spicules are kind of bending a little bit. Would you expect that to happen?
Well,
if there was a clatorizid?
Boy, I don't know. I mean, the spicules are going to be made out of silica, so I wouldn't imagine that they're going to be flexible. I mean, I don't think these spicules form like multi-spicular tracks. I think that, you know, I've just never seen anything like this, so I don't know.
Oh, wow.
Well, I'll certainly back you up on that, Shirley. Doesn't look like anything. And that pink certainly makes it look like tissue, but these just aren't tentacles.
Yeah, and you're sure about that, right? Because they're not flexible, they're not moving. Yeah, they're not moving around enough.
Nothing like this either before.
It's just very strange. Gosh.
No,
I'm gonna have our videographers give me a still of this later on, and I'm gonna have to send this over to Jean Vasselet and Nicole Boreas-No and have them take a look at it, because boy, this is kind of
very unusual.
Yeah, I mean, just the stalk looks kind of unusual to me. That pinkish color looks, it just doesn't look sponge-like to me. The only thing that looks sponge-like to me is like that central part where that, you know, the dandelion part is.
Yeah.
Okay. All righty. Well, this is very interesting. Yeah, I suppose so.
Josh, you see that thing in the overhead view that's broadside?
Keep a lookout for a small rock with a similar thing on
Something coming down this way. It looks like it's on the bottom.
I think that might be...
Is that just a trail or a rock? Or
might be just soil.
Soil, maybe.
Oh, no, that's a...
Oh, okay. That's you.
Yep.
Okay, yeah.
Oh, that's a scrape mark in the white on the right side of the screen from the...
Leaving my mark.
G2
from
was
the
here.
skeg of the ROV. But that's good to know, because then we know that the sediment right under the surface here is lighter, and very likely these little white patches are in fact from burrowing animals.
Bioturbation.
Yes, bioturbation. Actually, I'm taking a peek through the Deep Sea Drilling Project's Leg 60 core photos of the Mariana Forearc to see how much bioturbation there was in the sediment.
Come in, video, I'll do a pirouette. Just
sounds good. I'll hold a tightish partial.
Another brittle star.
See what else we have. Alright, come on out.
Something yellow to the right. Not sure what that is.
Come on in. It's a,
Okay, what do we got there? Oh, another barnacle.
Oh, it's got its legs
a
out.
cirri, stalk barnacle.
And then, is that a little sponge behind it?
Back in. Oh,
Where?
Yep.
Our pilot is asking if he sees a sponge behind. So carnivorous one?
That the rock that's just above it?
Yeah,
is that
come
you're
on in
looking
video.
at? Yeah.
Where?
Oh my gosh. It is.
Oh, what a cutie.
Oh my gosh.
Oh my, yeah, look at that.
That guy we could probably get.
Yes, you certainly could.
Yeah. That's
okay.
as cute
All right.
as it can be.
Okay. Okay, we're going to take a vote, except I have 10 votes. No, not just kidding.
Well, if we're going to take it, we have to decide relatively.
Okay, let's do it. Take it.
Yeah, ship's done with that move.
Okay, taking a sample
take it.
of
We have
a mini sponge.
We got
get your
45 minutes
hydraulics
left.
going
Take it. It's
It's
never
just
seen
that we're
anything like this
moving.
before.
All right, guys, I'm going to call another.
Yeah,
50
I agree,
meters, same
Scott. It's
direction.
not exactly the same, but
Nope.
Great.
It's pretty darn
so
close.
So you want to go for that other drawer in the port box?
Sure.
I feel like I've seen lots of that kind of clitorized sponge. I refer to them as lollipops, so the lollipops is fine. Whereas the other one, you know, had the ring of spicules in a different orientation, and then had that center raised part.
Well, what do you think? You want to wait? You want to wait and just move forward because there's some that looks like there's. All right, we can just take a real close look at it.
No, I'm not saying to wait. I'm just saying I think that it's different from the one we were looking at before. The one we were looking at before, I think, would be really exciting to collect, but I'm not telling you that you shouldn't collect this one.
Okay. You know what's going to happen, of course, Scott. We're going to see one like we saw before, like 10 meters from here.
Absolutely. Absolutely. This one seems so small. I'm not sure how you're going to pick that up.
They're going to pick up the rock.
Maybe.
So are we going for it?
I can't even tell what rock it's on.
Okay, what's the verdict? Go for it. Yeah, go ahead, pilot.
I guess my vote might be changing my mind a little bit here. Maybe
stand by,
wait and see if we can find that weird one again with the pink stalk.
Okay, we're going to wait and see if we can find the weird one again with a pink stalk.
Serial should be stopping.
Okay.
Okay, so this is a no-go, huh?
This one's a no-go.
Hey, let's come in partial
Okay,
video.
that's fine.
Yeah,
that's okay.
understood.
Because
if
I just know we're going to see the other one.
You hope,
And
yeah.
I'm going to be kicking myself.
Well, there are
There's
a lot of
something
rocks in this part
something white and
of
small.
the top of the slope,
Red
so
ROV
if that guy
might be
needs to
something
have a
just
rock
hydraulic
to
secure.
attach to,
50 meters, bearing 0403.
then the chances are better.
0.1 knots.
Further to the right.
I gotta push ahead.
Yeah,
Come on out.
sounds good.
Yep. Oh, there's a crab
Skip the
walking
crab.
along. Yep,
Good copy,
just
thank you.
Zooming crab.
quick. This is something different. This is a little crab we haven't seen before. Oh my goodness.
What's it got on it?
It's got something on it.
Anemones. Looks like it's got some anemones.
Is this a squat lobster with an anemone on the back, the different
like
sort
a few of them.
than we've seen before? I think that's
It's
what it
one of
It has
is.
a very painful case of zoanthus.
It does.
So we are seeing more critters. I guess maybe that's because we have a harder substrate with all these rocks here.
Of course, with only 40 minutes left
Yeah,
in
if
the
we're not
dive.
moving,
I
are we?
know.
Ship stopped, right?
Ship is stopped, but
We're still,
See, that's
yeah.
what happens if you don't remove the
looking straight
polyps
down
in that
on it.
region of your body soon enough.
Yeah, I probably called that movie way
Chris!
too early.
Okay, we are not going to continue on this line of discussion
Nope.
because the temptation is just going to be
Too much.
too much.
Okay, fewer rocks now. So that just was area.
Get some ground.
Ooh, we're
and go look at
racing
this.
across the bottom now.
Just pushing out ahead, watch Lee.
Yeah, we're pushing out ahead so we can get ahead of Sirius.
Outcropping.
Just come in here.
Fragments of rock, possibly.
A little bit.
And she is wondering, this is different. It is really hard to tell whether these
I do
are
see tiny
hard
little white
rocks
spots here
or
and there.
just covered with
Yeah.
manganese.
Hard to tell what they are, though.
Yeah, come on out.
They're
Sorry,
certainly
what'd you say?
Yeah, come on out.
not obviously in place. Not too helpful for geology if they're not in place.
Really helpful for biology, though.
Yeah, I know what you mean. Yeah. Making little display stands for the corals here. Not corals, sponges. Sorry.
Corals too. You never
Yeah,
know.
corals.
You never know.
Well, we have seen,
A dead bamboo coral.
we saw,
Zoom in
and
here,
that
video.
big stalked thing. Okay, what's
Oh, we have a
this?
couple things coming up.
Okay. Oh, surely, that's a sponge. I'm sure that's a sponge.
Now see this?
All right,
It's
come on,
different.
video.
This to me looks.
Yeah, but
This
the
is a
one
weird
on
one.
the lower left
You
looks
think
like
it
the other one, Cheryl.
I think it is. Is that rock too big to grab the whole thing, or
No.
Because that's
Wait.
going to be really hard to get the animal.
Wait, are you?
That rock
are you,
would be an outstanding collection.
Well, look, it's only
wait,
like
are you the
12
one right in the center?
centimeters.
Are you looking at the one right in
Yeah.
the center of the screen?
Now
Is
look
that
at
the
the
one? All right, let's
animals
look
on
at
it.
the animal on
All right,
that.
come on in, video.
Because there's something there, yeah.
There's
it's
several
to the
animals
left.
on
So
this.
there's a hexactinellid at the top.
But the
and
thing
then,
on the left is the same
Is
one.
that the one?
Yes,
that
it is.
Yeah.
Oh, grab it, Shirley.
Oh my God, there's all kinds of stuff.
Oh,
Okay.
and there's a,
Oh,
yep,
it says
there.
Oh my God, there's the other Cladorhiza there.
Yeah.
Oh my gosh. Okay. All right.
We need this rock.
Yeah, go ahead, pilot.
We still have 30 meters in the smooth, so I'm
Okay,
probably
and
gonna
in a
ask them to
bio
hold
box.
Yeah.
Here.
Yeah, you can do that.
Deb goes, and although no one else cares, she likes the rock. We're gonna have to.
Current position.
We're gonna have Matt make several commensal labels for this one.
All right, let's
Oh,
see
and
if
Deb, I
this
know
will
you're going to be.
Really,
Yes.
it'll be interesting to see if the amphipod that was on the back Cladorhiza is still in that same position when you
I
get it up in
know.
the lab.
I know.
Pilot,
I know.
just take care
'Cause there are
and
like
be
they're the
be
two
gentle with this, because if
Cladorhizas.
this rock is if it's a
yeah.
siltstone, it may crush like the earlier one did.
Did you
That's
see
a
there
good
was one
point.
of those
Thank you.
amphipod sticks on there too?
Calm down, calm down. Trying to shut her up so I can tell the pilot the rock might break up. Okay, so we want to grip it gently on the non-biologic side.
It's at least consolidated enough for these animals to attach to it.
It's definitely loose. Yeah, definitely loose. Really good angle to grip it. Oh, perfect. I suggest immediately putting it in a bio box. Immediately in a bio box?
Okay, when you're ready, Don.
Ready? Drawer open.
Where are you thinking of putting this?
That was the port side bio. Inboard.
We have something in port inner.
We have
can
inboard.
go
Oh, you had it?
Port outer.
You have port. You have
Go
port
port
inner.
outer.
Port outer, okay.
Yeah, you already have something in port inner.
Sponge and all kinds of stuff.
Do I have anything in starboard inner?
Starboard inner is available. Starboard outer.
You want to go there?
Yeah, we're going to
All
starboard.
right. Closing drawer. Okay.
The rock sample.
Retracting
Fractal rock.
the pin there. And extending the pin on the starboard side.
That's where we put it.
Time. I got the time.
Okay. Opening drawer.
Hang on there, critters. Gently, gently, catchy monkey. Ooh, the nip is shaking around here. It's
I got some feedback on the
yeah.
there. I'm very sorry for that.
No, understood.
Watch leads nav. We're calling this a biosample, right?
We are now.
Great.
And we got it.
Very
Oh,
good.
thank you, pilot. Great
Nicely
job.
done.
Close.
Okay, closing drawer.
That is
right.
awesome. Thank you.
Are these guys good or what? No wonder they have groupies on shore. They have groupies on the ship, too. Wow, I know. Scott Francis. Yeah, Scott France said tremendous collection. Just, you're getting all kinds of accolades from the shore. Very impressed.
Thank you, Scott.
Hydraulic secure.
Hey, thank you. That's why I generally don't use force feedback, because occasionally you get those oscillations, and it
what
always
causes
happens.
Just noise coming back down the
Right
line?
when you don't want it.
Oscillation between there's a delay between the arm moving and the mini master moving, and you go back and forth, and it amplifies. So you can adjust the gains to get rid of that, but there's a certain point where if you gain down so much, there's a lag in your motion. So we can start moving again now.
Okay, he's out far enough ahead, or
yeah, we're ready to go.
Okay, great.
So I generally just run without force feedback.
Are we right up on top now? Should I stop?
We are.
Yeah, we're good. We're on top.
Yeah. So I'll do 020.
Yeah, Nav, how far are we from dive in?
Come on in, video. And it always happens.
We are about
At the worst moment, that's
the
right.
most delicate sample.
Beautiful.
And you almost got it in. It just.
Oh, good close-up on that brittle star.
That's when
Now,
you're trying to make your
this is the
finest
same one we
movements
saw
as well.
before, but now its
usually
arms are up to collect
it gives you feedback when you're
food
up
from
against
the water,
stops.
right?
Uh-huh.
So it'll
Yeah, I
stop
think
the mini
this is
master,
a
and if you,
brittle star.
that's
right?
when you can
No,
get
I
an
think that
oscillation.
This is a sea
So
star?
it's usually right when you're like
I'm wrong.
It's a
Yep.
sea star.
right in on.
That's
It has
right.
6
Bridge
legs.
RV nav.
Oh, and look, there's like a gelatinous
Like
white
another
thing
move,
up at the top of the
50
Yes,
screen.
there's another
meters,
clatterized sponge over to the
0
left.
0.19.
Well, that's good. We got one.
Yes, it is.
It is a sponge.
Is that another sponge?
A
in the
sponge?
background?
Yes, it
No,
is.
in the
Thank you.
background, I think that is at some
Okay,
sessile
co-pilot,
animal.
this mode is gonna be at
What do they call those
020.
things?
020, copy.
It's
That's
one in the top
Are you
right.
looking for tunicate? The word
No,
tunicate? Is that what you're
Tunicate,
looking
or I don't know. If maybe it is a sponge.
Yeah,
It does
I
look
don't
like
know.
it's got texture on the surface, but I thought maybe it's some like
for? Yeah, I
little
was distracted.
holothurian
I was
or.
distracted by the sponge.
Of course you were. We
Yeah,
understand.
no, I can't tell.
Well, we have to zoom on it. So, pilot, could we zoom
Yeah,
when you're
Scott-
done
Scott,
on the
yeah, I
white jelly thing in the back, just
think
above the
that's
arm
a
of
sponge.
everything?
It might be a sponge.
I don't know. Oh, we're gonna settle down and get a little closer
Go ahead.
so we can zoom in on all the critters. Okay, folks on shore. What is this? When it comes into focus,
Sponge.
it's a sponge. Shirley was right. Not surprising. Good enough?
Perfect. Yeah, that's good.
Thank you, pilot.
Okay, let's go find a coral.
We want to go find a coral, apparently.
Another one of those crabs to the left.
Oh, yeah. Crab to the left.
It better be attached to a rock.
Oh, I see the crab.
Come on in, video.
Well, it'll be interesting to see if this
take
one
that control if you
has.
want.
No, I'm just going to leave it because I think they're going to want another sample.
Oh, wow. No, what is that on? What is that waving around in the rock, Shirley?
I don't know. Is it a hydroid?
Yeah, it looks
I
like
don't know.
it.
Can't tell.
Then there is a star, a brittle star, I think, to the right. Oh, that's a pretty hydroid, though. Little white arms with white dots.
I have a bit more zoom,
And
too.
I think there's a brittle star to the right on the seafloor.
Come on down a little bit, video. Yeah.
Oh, there we go.
All right, come on in.
Boy, as we zoom in, we can see all these pebbles really nicely too. I'll be interested to see what the rock is and whether it is sedimentary. I'll let you folks know tomorrow. Tune in for the diagnosis. You see the brittle stars start to move. They really can go fast. I was so impressed. Several of the zooms we've done on brittle stars, they've just scooted out of the way.
Okay, when you're good.
Thank you, pilot. That's great. Go ahead.
Moving along. Something on the rock over here.
Yeah, there's white something sticking off that rock. Big one. We got another one here, Shirley.
Yeah, I know.
They're everywhere. Well, that's a good point.
That's good. Yeah.
That there are a lot of them. So we know that they are perhaps characteristic of this region.
This looks like it's a